Greetings of the day. I am Professor P. Saujanya, Professor and HOD, Department of Pharmaceutical Biotechnology, Vignan Pharmacy College, Guntur. I wish to describe about identification of bacteria by biochemical test. Bacteria has been identified by staining methods and biochemical tests. Biochemical tests are used to identify the metabolic activity of organisms. To identify metabolic activity of organisms. So based on their metabolic activity, the bacteria has been differentiated. And the first test is carbohydrate fermentation test, also known as sugar fermentation test. And this test is used to determine ability of microbes to ferment the carbohydrates or sugars with the production of acid and gas. When the test organism is inoculated in sugar containing medium, the fermentation of bacteria is detected by the production of acid or by the production of gas, or both production of acid and gas. So here, if the fermenting bacteria are grown in the culture medium containing carbohydrate source, and they may produce organic acid as byproduct of the fermentation. And this byproduct has been detected by the addition of the indicator phenol red or bromocresol blue. And this production of acid is detected by the change of the color of media to yellow color. Yellow color indicates positive to acid production. And the gas production is detected by inserting Durham's tube in inward position in the culture medium. In the Durham's tube, if gas bubbles has been observed, that indicates the positive test. So, the test organisms which are able to change its color to yellow and produce gas bubbles in Durham's tube are positive organisms. If there is no change in the color of the medium, then that is negative organism. And here you can see three test tubes. One is uninoculated medium and the other two are inoculated medium. In the second tube, you can see the change of the color of media to yellow. That indicates positive test. In the third tube, as there is no any change of the color of the medium, it indicates negative. Next, indole production test. Indole production test is to determine the ability of bacteria to degrade the amino acid tryptophan. So here, the bacteria which have tryptophanase enzyme, they are able to degrade tryptophan into indole. Along with the production of pyruvic acid and ammonia. So this indole production has been detected by the addition of para-dimethylamino benzaldehyde. It is a ingredient in COVAX reagent. And this has been detected by the change of the color of media to cherry red color or quinoidal red violet color. So the development of cherry red color indicates positive test. If there is no any change in the color, that indicates negative test. So that means those organisms will not able to produce tryptophanase enzyme, so they will not convert tryptophan into indole. And here you can see the positive and negative organisms. In the first tube, you can see the change of the color of media to pink. At the surface, you can see the pink color. And in the second tube is negative. There is no change in the color of the medium. The methyl red test. So methyl red test is to differentiate Escherichia coli and Enterococcus aerogenes organisms. When these organisms are inoculated in glucose containing medium, if they are able to produce acid, and this acid production is detected by the addition of the methyl red. So here E. coli organism is able to produce three different types of acids: lactic acid, formic acid, and acetic acid. So after addition of methyl red, it changes its color to red. That indicates positive test. Whereas Enterococcus aerogens organisms, when it is inoculated in a glucose medium and after incubation, it produces a little amount of acetic acid. So when addition of the methyl red, it produces 2,3-butane-diol. So the color of the medium is yellow, hence it is negative test. The first one is positive test. E. coli is positive, and the second tube is there is no change in the color of the medium and it is negative. Enterococcus aerogens is negative. Escherichia coli is positive to methyl red test. And Vox-Proskus test is to differentiate Escherichia coli and Enterococcus aerogens by the production of 2,3-butane-diol and acetone during the via glucose fermentation process. So when these organisms are inoculated in glucose medium, if they are able to produce acetyl 2,3-butane-diol during the production of acetyl methyl carbinol. So this 2,3-butane-diol is an intermediate compound during the production of acetyl methyl carbinol. So the production of 2,3-butane-diol has been detected by the addition of alpha naphthol. So after addition of the alpha naphthol, if the color of the media has been changed to pink by the production of diacetyl and creatinine, that indicates positive. So the development of crimson red color indicates positive for Enterococcus aerogens. Enterococcus aerogens here is positive. And if there is no any change in the color, that is negative. So E. coli is negative here. So these are the two positive and negative samples. So the positive is Enterococcus aerogens and negative is E. coli. No change in the color of the medium. Citrate utilization test. This citrate utilization test is used to determine the ability of organism that uses citrate as a sole source of carbon. So the organisms which uses citrate as a sole source of carbon are positive to citrate utilization test. And the organisms which cannot use citrate as a source of carbon, they are negative to citrate utilization test. So when the organisms are inoculated in sodium citrate medium, the medium containing sodium citrate, then the organisms which have citrate permease or citrase enzyme, citrate permease or citrase enzyme, they will use this sodium citrate as a source of carbon and thereby they produce pyruvic acid, oxaloacetic acid and carbon dioxide. So the utilization of sodium citrate as a source of carbon is detected by bromothymol blue indicator. Bromothymol blue indicator. So if the media color is changes its color to blue, indicates positive. No change in the color indicates negative. So here you can see when the organisms are inoculated in Simon citrate medium that containing sodium citrate as a source of carbon. The first tube is an inoculated medium and the second tube is positive test because it changed its color to blue due to the presence of bromothymol blue. And the third one is negative because there is no change in the color of the medium. And the positive organism here is Enterococcus aerogens. Next, nitrate reduction test. And this test is to determine the ability of microbes to reduce nitrate to nitrite. Nitrate to nitrite. So, the organisms are inoculated in nitrate containing medium. That is potassium nitrate, 0.5% potassium nitrate containing medium. If the organism possesses nitrate reductase enzyme, it reduces nitrate to nitrite. And the nitrite accumulation is detected by addition of sulfanilic acid and alpha naphthite amine. So this changes its color to red with the production of sulfobenzene azo alpha naphthide amine. So the red color indicates positive, no change in the color indicates negative. So the first tube is positive, change of color to pink, and the second tube is negative. Next, urease test. Urease test is to determine the ability of microbes to degrade urea by urease enzyme. So the microorganisms which have ability to produce urease enzyme, it will degrade this urea into ammonia. So urea is a diamide carbonic acid, often referred as carbamide, and this hydrolyzes urea to ammonia with the production of a specific enzyme, urease enzyme. And this has been detected by inoculating the test organism in Christensen's urea medium. And this medium contains the pH indicator phenol red. So here you can see the urea has been reduced to ammonia. So when the urea has been reduced to ammonia in presence of urease enzyme, so due to the accumulation of ammonia, it causes alkaline nature of the medium. So due to the alkaline nature of the medium, the indicator color has been changed. So the color changes from reddish pink to deep pink indicates positive test, and no change in color indicates negative test. So this is urease test. In a Christensen's urea medium, the test organisms has been inoculated. The first tube is uninoculated medium and the second tube is positive. So changes its the color from light pink to that of dark pink. So here the organism whatever has been inoculated is able to produce urease enzyme, and the third tube is negative. There is no change in the color. So these organisms are not able to produce urease enzyme. Next, triple sugar iron agar medium. So triple sugar iron agar medium is used to differentiate the bacteria under Enterobacteriaceae family. This family contains multiple organisms. To differentiate each organism, this triple sugar iron agar medium is used. So here the media contains triple sugars like 1% concentration of lactose, sucrose, and 0.1% concentration of glucose along with pH indicator phenol red. And this phenol red is incorporated into the medium to identify the production of acid from carbohydrate fermentation. The uninoculated medium is red in color due to the presence of phenol red dye. And along with these sugars and indicator, the media also contains yeast extract, beef extract, peptone, then sulfur, trace elements, and vitamin B complex, etc. Along with the sodium chloride to maintain osmotic equilibrium. So then it also contains sodium thiosulfate and ferrous sulfate, which makes hydrogen sulfide indicator system. And as well as this thiosulfate is reduced to hydrogen sulfide by several species of bacteria, and hydrogen sulfide combines with and form insoluble black precipitate. So the black precipitate indicates the production of hydrogen sulfide, and the blackening of colonies indicates the production of hydrogen sulfide. So after inoculating the test organism in triple sugar iron medium, it has to be incubated for 18 to 24 hours to detect. Here three tests together has been detected. They are sugar fermentation test, gas production test, hydrogen sulfide production. Sugar fermentation is detected by pH indicator, change of color of media to pink. Gas production is indicated by inserting Durham's tube in the medium. And hydrogen sulfide production is detected by change of the color of colonies to black. So the indicator is pink at alkaline pH and yellow at acidic pH, and neutral pH it remains red. So these are the following reactions that may occur in the triple sugar iron medium. So if the formation of yellow bud and red slant has been observed, yellow bud and red slant. Yellow bud means growth of organism is yellow and the slant will remain as red. That indicates that due to the fermentation of glucose, phenol red indicator turns yellow due to the persisting acid formation in the bud. And the slant remains red due to alkaline nature because of limited glucose in the medium and therefore limited acid formation which does not persist. So the alkalinity of the medium makes it remains as red. And if the medium has been yellow and slant, if the tube whatever has been observed is yellow bud and slant, slant is colorless. So slant and bud is yellow. So yellow because due to the fermentation of lactose and sucrose. So that causes the yellow color colonies. And that leading to excessive acid formation in the entire medium. And gas formation. Gas formation is noted by splitting of the agar medium. So the splitting of agar medium, the gas here formed is hydrogen sulfide gas. So the gas formation is detected by splitting of agar medium. Whether the gas formed is hydrogen sulfide or not is detected by changing the agar medium as black color. Blackening of agar indicates gas formation. And if the medium is red bud and slant is also red, red bud and red slant, that indicates that none of the sugars were fermented because the red color is due to the indicator. It is not changed to yellow. That means the sugars has not been fermented. And the slant is also quite stable. That means neither gas nor hydrogen sulfide gas has been produced. Red bud and red slant is negative to three tests. The three tests are sugar fermentation, gas production, and hydrogen sulfide production. Sugar fermentation is detected by change of the color of media to yellow. Yellow indicates positive to sugar fermentation. Gas formation positive is splitting up of the agar medium. Hydrogen sulfide positive is by blackening the color of the agar medium. So here this is the interpretation of triple sugar iron medium, alkaline slant or acidic but only glucose is fermented. If acidic slant or acidic but glucose, sucrose, lactose, all three sugars are fermented. Bubbles or cracks are present that indicates gas production and black precipitate present indicates hydrogen sulfide production. And here you can see the differences between the different organisms, slant, bud, gas and hydrogen sulfide production. Hydrogen sulfide production has been observed in the tube C and the tube D, blackening of colonies. And then gas production has been observed in second. You can see the breaking up of the medium in the second tube. Next, oxidase test. Oxidase test is used to determine the ability of microbes to produce oxidase enzyme. So this oxidase enzyme plays a key role in electron transport chain during aerobic respiration. So here cytochrome oxidase catalyzes the oxidation of reduced cytochrome by molecular oxygen, and this results in the formation of water and hydrogen peroxide. And this test is positive to aerobic and as well as some facultative anaerobic and microaerophilic bacteria. They show oxidase activity. So here in the cells, if the reduced cytochrome has been oxidized to cytochrome C. So due to the presence of cytochrome oxidase, that indicates positive. So here in the cells, you can see the purple spot. In tube, how you can detect the oxidase test? So this oxidized cytochrome after addition of para amino dimethyl alanine oxalate. After addition of para amino dimethyl alanine oxidate, it causes reduced cytochrome C, and the color of the medium has been changed to blue or dark purple. And here we can see the development of dark purple color after the colonies has been added. So this oxidase test is positive to all aerobic, facultative anaerobic and microaerophilic bacteria. So once this filter paper has been moistened with oxidase reagent, and if we place the colony at the center, the change of the color of the organism to purple indicates positive. Next, catalase test. So catalase test is used to detect the ability of bacteria to produce catalase enzyme. So there are certain organisms which produce hydrogen peroxide during aerobic respiration and sometimes extremely toxic superoxide radicals. This presence of superoxide radicals causes destruction of the cell constants, and thereby this is toxic to the cells. And whereas aerobic bacteria possess an enzyme like catalase which convert this superoxide radical into the water and hydrogen peroxide. So the bacteria must be able to protect itself against such oxygen products or it will be killed. So many bacteria possess enzymes that afford protection against this toxic oxygen products. So here, obligate aerobes, facultative anaerobes, usually they contain enzyme superoxide dismutase, which catalyzes the destruction of superoxide radicals. So this superoxide dismutase enzyme will neutralizes the superoxide radicals, and either catalase or peroxidase catalyze the destruction of hydrogen peroxide. So, the catalase production and activity can be detected by the addition of adding the substrate hydrogen peroxide. Hydrogen peroxide. So, this hydrogen peroxide with the catalase enzyme will causes bubbles. So, if a catalase was produced by the bacteria, they will liberate free oxygen on the reaction. Liberate free oxygen on the oxygen gas on the reaction. So the bubbles of oxygen represent positive catalase and the absence of bubbles formation indicates negative catalase. So the aerobic organisms, facultatively anaerobic organisms, microaerophilic organisms have this catalase or peroxidase enzymes to reduce the accumulation of superoxide radicals by superoxide dismutase enzyme. But anaerobic bacteria doesn't have this enzymatic system. Superoxide dismutase or catalase or peroxidase. That is why oxygen is toxic to anaerobic organisms. So here you can see the formation of bubbles on the first tube. So it is positive. Notice the bubbles. So positive is Staphylococcus aureus catalase positive. And the next one, no formation of the bubbles, Enterococcus faecalis. This is negative. Next, starch hydrolysis test. Starch hydrolysis test is to determine the ability of bacteria that hydrolyzes the starch. So the bacteria which have hydrolase enzyme will hydrolyze the starch into oligosaccharides and monosaccharides. It splits the polysaccharide starch into smaller molecules in presence of water. So this starch contains two constituents, amylose and amylopectin. Amylose and amylopectin. And both are rapidly hydrolyzed by certain type of bacteria using their alpha amylases and to yield dextrin, maltose and glucose. The starch has been degraded into dextrins, maltose and glucose by alpha amylases enzymes. So here you can see the starch has been hydrolyzed into dextrin, maltose and glucose. So the large polysaccharide has been broken into the intermediate polysaccharides and disaccharides and monosaccharides. And how this starch hydrolysis has been detected. So once the test organism is inoculated in starch agar medium, after incubation, if you add grams iodine, starch with iodine gives blue color. So the blue color indicates negative. That means the starch is not hydrolyzed. If the starch has been hydrolyzed, then after addition of the grams iodine, it will not show any color. That is negative test. If a clear area appears after adding grams iodine to the medium containing starch and bacterial growth, then the clear area indicates positive to starch hydrolysis. If there is no clear area, starch has not been hydrolyzed. Thank you for watching.